Hello, and welcome to biology. So, today I want to talk about cellular reproduction. And when we talk about cellular reproduction, we're talking about how to make more cells. In other words, mitosis, which is asexual cellular reproduction. And the first question, of course, we have to ask is, what's the point? What is the point of mitosis, which is this cellular reproduction? Is it the reproduction of the organism, growth, regeneration, repair of cells? And so this question is actually asking which is not a result. So 3 of them are. Well, it turns out that reproduction of the organism itself is not the goal of mitosis. In fact, mitosis is for growing, right, for regenerating parts and for repairing cells, right? So that's why cells divide. And think about if you're injured, you're going to have to repair it. You go through puberty, you get bigger, right? You actually gain mass as you grow. And so all of these things, this is what mitosis is for, okay? So when we talk about mitotic or mitotic, you can say it either way, cell division, we are talking about the division of somatic cells only, okay? Those are the non-reproductive cells. We will be talking about the reproductive cells, but that's a different process called meiosis. Now, this is going to be in your eukaryotic organisms that we're going to be talking about. And what you have is basically the cell copies itself. And so you get 2 that are identical to the original one, right? So the process is really important in terms of chromosomes. And I've got here maintains chromosomal ploidy. Well, what that means is that if you start with 46 chromosomes, you have to have 46 in the end, okay? And so you want to maintain, because remember, ploidy is the number of chromosomes that you have, okay, or sets of chromosomes. So we talked a little bit about diploid versus haploid, but I want to really make sure you understand that. The haploid cells are the gametes, the egg and the sperm. And we call the number of chromosomes that each one has N, right? And so they're N chromosomes. So in humans, there are going to be 23. N is equal to 23, right? In other things, it's going to be other numbers. All of the rest of your cells besides egg and sperm are diploid, remember? And so they have 2 copies, a pair, of each set of chromosomes, or of each chromosome, and so their number is 2n. So a diploid cell has 46, 2 times 23, okay? And that is going to be maintained in mitosis. And again, we'll talk about meiosis later on. Okay, so when we talk about the eukaryotic cell cycle, this is going to be a series of stages that the cell passes through, starting with the single cell and ending up with the 2 daughter cells, okay? Now, in single-celled eukaryotes, this is how they are going to reproduce, and that is, of course, asexual reproduction. There are some multicellular eukaryotes that also asexually reproduce. We'll talk about that a little bit later on. But in multicellular eukaryotes, typically this is going to be just cellular reproduction in general, but if you only have one, you're kind of making copies of yourself. So what is in the eukaryotic cell cycle? Lots of things, actually. There's lots and lots of things going on, all kinds of checkpoints and signals and, you know, all of this good stuff. But you can really break all that up into 3 events. There's interphase, and during interphase, it's all about replicating the DNA, okay? And of course, the metabolic activities of the cell are going on and all this good stuff. But DNA replication is happening during interphase. In mitosis, this is actually the process of division. And then cytokinesis, which is not part of mitosis, it's its own thing, is where the cytoplasm is dividing and you get new cellular membranes. It's like I said, it's not a phase of mitosis, it's an event in the cell cycle. Okay, when we look at these events, we can kind of divide them up into these 4 stages, right? We have the gap 1 stage, and that is before chromosomal replication. It is a part of interphase. Then we have the synthesis phase, okay? And this is where the chromosomes are being replicated, right? This is the DNA synthesis. This is also part of interphase. And then there is the gap 2 stage, and this is the period right after chromosomal replication, which is part of interphase as well. Now, in the gap phases, it seems like there's nothing going on, but there's actually a lot of stuff going on. You know, there are, like I said, there are checkpoints and things that have to happen before the cell can move to each different stage of the cycle, right? And so we're not going to talk about those, but I do want you to understand that that's true. I mean, there are lots of mechanisms where the cell can actually stop cell division if things are not going well at any point in the cell cycle. Okay, and then the last part of this stage is going to be the M phase, which is mitosis. All right, and this is where you can see all of these kind of together. Okay, so interphase we already talked about, but if you're looking at a cell in interphase, you're going to see a well-defined nucleus. You're not going to see individual chromosomes because they are still in the chromatin phase. This is all the uncondensed, right? It's like the inside of a golf ball, like tangled up stuff. And then you'll also probably see a nucleolus within that, cluster of chromatin, which is the DNA. Remember, the nucleolus is the site of RNA synthesis. Again, you may or may not actually see that, but it's probably there. All right, and this is actually what the cell looks like. It looks like just what you think about when you think about a cell, because it spends most of its time in interphase anyway. Now, during mitosis, the chromosomes are going to condense. You're going to actually be able to see them. You're going to have the chromatids separating, right? And I have a question for you about what a chromatid is in just a second. And then you're going to have those being the daughter chromosomes. They're going to move, and then you're going to have the cell actually split. So let's talk about the stages of mitosis. The first stage or phase, you can call it whatever you want, is prophase. And this is where the nuclear membrane is going to disperse, right? It's going to break down, and the chromatin is going to condense, and you're going to get these nice little chromosomes here. They're visible. They look like that nice pair, little X thing. And then the mitotic spindle apparatus begins to form, and that is going to be what is vital. You're seeing here, this is the mitotic spindles, right? And so you've got the cell nucleus with the chromosomes and all this good stuff. So they're going to be vital in actually pulling the chromosomes apart, which we're going to talk about in a second. But here is your question. What is a chromatid? Well, the chromatid is 1/2 of the chromosome, right? And so the chromatids are going to separate. So you've got two sister chromatids in every chromosome during mitosis, okay? All right, now, after prophase, you've got all these great little chromosomes. They are going to line up in a nice line here along the equator of the cell, the middle of the cell. And you can see here the spindle apparatus, which is really cool, actually. And you can see the centrioles here. It's very exciting. And this is actually another one where you have a different coloration process, but it's very cool. So these are going to be like the microtubules, and here are the chromosomes. Wow, I don't know why I couldn't think of that. Okay, now anaphase is the next phase. So prophase, you're condensing, and you're dissolving the nuclear membrane. Metaphase, you're lining up in the middle, middle, right? And you're going to have that nice line. In anaphase, they're going to move away from each other. So this is where the mitotic spindle apparatus, okay, is these microtubules are going to shorten. They're going to pull the chromosomes away from each other, right? And this is good because now you can have a nuclear membrane form around each of these and get two new nuclei that have all the right numbers of chromosomes. That's very exciting. And that is what happens in telophase, right? Telophase or telophase, you can say it either way. So the chromosomes at this point have moved to the poles of the cell. They are going to decondense, and you can see it here, right? They're going to decondense, and they're going to return to that state of chromatin. So they're going to tangle up and look like they normally do. And at this point, you're going to have two new nuclear membranes and nucleoli, right, the nucleolus, form at each of these spindle points, okay? And so now you have two new identical nuclei. So the cell division inside the cell is done, okay, for the most part. By the way, all of the organelles also replicated. So at this time, you're not just replicating the nucleus, you're replicating all of the organelles. And that's important to remember too, because the new cell is going to need all that stuff as well. So the cytoplasm, the mitochondria, ribosomes, all of those things, those are all replicating as well, okay? Endoplasmic reticulum, Golgi apparatus, all that. So here's your next question. Which sketch shows the arrangement of the chromosomes that you would expect to see in metaphase of mitosis for a cell with a diploid chromosome number of 6? Well, uh, this this one, right, has nothing to split. And so we know at this point in mitosis that you have to have pairs, right? They're all lined up in pairs. Well, B and through D are pairs. This has 6 chromatids, but it has only 3 chromosomes, and we want a diploid chromosome number of 6, okay? And then in this one, you need to have one copy, right? One chromatid from each chromosome in the new cells, right? Here are your little centrioles, by the way, and this is the spindle apparatus, just as an aside. Well, this has 6 chromosomes, but this is going to be very unevenly distributed, right? And that would be bad in this case, okay? But here, look at this. They're all lined up nicely, and when these start to pull apart, you'll get 1/2 of each, right? You'll get 1 chromatid from each one. And that's very exciting. So D is the answer. Okay, now let's talk about cytokinesis. So we've had the nucleus, right? We had all of that replicate during mitosis. This is the final phase of the cell cycle, cytokinesis. Remember, it is not part of mitosis. But what you have is the cell membrane or plasma membrane starts to pinch in the center, and this kind of like a ring, like a twist tie almost, condenses, right? It contracts. It's called the contractile ring because of that. And so it contracts and it pinches it off, right? And you get 2 cells. And this is going to begin at the end of anaphase, and it actually marks the beginning of the telophase. So cytokinesis and telophase are happening at the same time, right? And this is what it looks like. You've got this little ring here, and it's started out like this, and then it got, you know, smaller and smaller and smaller and smaller until these pinch off. Now you've got 2 new, beautiful, identical cells. Now, in plant cells, it's a little bit different because you have that rigid cell wall. And so you can't just squish it and pinch it off. And so in this case, instead of getting that contractile ring, you get a cell plate, right? And so you can see here that a new membrane and a new cell wall are forming in the center. And so this is going to start in the middle and then spread out to either end until it is complete and you have 2 new cells. And then this is the whole system all together, right? Now, the new daughter cells, they're an interphase, right? And so we're going to pick up with this daughter cell here. Now it becomes the original, the parent cell. And so here we've got all of our things. Our centrioles are forming, right? We're just doing, their centrioles are just hanging out. We're doing all these new things, right? Here's the nucleolus. We're doing normal cell stuff, okay? As we start prophase, we start to condense. Oh, and by the way, this was the S phase where all of the DNA is being synthesized, replicated, okay? Then we move into our prophase where we're condensing, and then, by the way, prophase is actually divided up into 5 smaller phases. You do not have to know that, but if you are doing any reading outside, you may see that, but we're just concerned about prophase as a whole, okay? So they're going to condense, the spindle fibers are going to attach, right, to the centromeres of the chromosomes. In metaphase, they're lining up. In anaphase, they're being pulled apart, right? Pulled apart. And then in telophase, you're getting a new nuclear membrane. They're decondensing, and cytokinesis begins at the end of anaphase and then goes through telophase until you have 2 new cells. And then this I put in here later when you're reviewing for your, I don't know, say test or something. This one is going to show you the difference between mitosis and meiosis. And meiosis is going to be our next conversation about sexual reproduction. Okay, so that is it for this. If you have any questions, please ask me in class, and I will see you next time. Thanks for listening.